Good morning to everyone. My name is Paolo Stincone, and today I'm going to present you my qualification exam titled Antimicrobial Peptides against Listeria monocytogenes proteomic approach and potential biotechnological application. My academic tutor is the Professor Adriano Brandelli. Because of pathogenic microorganisms, every year 600 million of people fall ill after eating contaminated food, and unfortunately 420,000 die. Listeria monocytogenes is one of the more than 20 species of Listeria, and listeriosis is the disease caused by the action of this pathogenic microorganism. Fortunately, is a relative rare, but it's severe because it's causing height, hospitalization, and mortality rate. Listeriosis signs and symptoms are fever, nausea, muscle aches, and diarrhea. And people with higher risk to contract this disease are pregnant women, older adults, children, and people with weakened immunity. Listeria monocytogenes is a foodborne pathogen, is an anaerobic gram-positive bacteria, and it's have the capacity to survive at low temperature, low pH, and high salt concentration. Because of this characteristic, this bacteria can survive in food not previously pasteurized, such as soft cheese, processed meat, smoked fish, fruit and vegetables. To overcome the problem of the eventual presence of pathogenic microorganisms in food, the industry use antimicrobial peptide. In this way, the shelf life of the product is improved. The antimicrobial peptide, in general, it's possible to be divided in two main families. Not ribosomally synthesized antimicrobial peptide and ribosomally synthesized antimicrobial peptide included in the group called bacteriocin. In this work, I'm going to use a group of compound not ribosomally synthesized, such as cyclic lipopeptide and nisin and leucine, that are two groups that making part of bacteriocin, so the ribosomally synthesized peptide. Nisin is a commercial antimicrobial peptide and is used in the food industry since 1988, when the Food and Drug Administration considered this antimicrobial peptide as a GRAS additive, and today is used in more than 50 countries around the world. This antimicrobial peptide is constituted by a polypeptide of 34 amino acid residues. It's secreted by Lactococcus lactis, subspecies lactis, and is have the ability to inhibit gram-positive bacteria. This last decades, the large use of nisin have been led to the emergence of antimicrobial resistant in gram-positive bacteria, including Listeria monocytogenes. That's why the need of new antimicrobial peptide is becoming always much more important. In this context, a group of Bacillus species has been isolated from freshwater fish goat coming from Amazonian rivers. Among this group of Bacillus, Bacillus p34 was considered as a good producer of antimicrobial peptide. Bacillus strains are known to produce three main family included inside a group of the cyclic lipopeptides. These family are named surfactin, iturin, and phenycin. These three family are characterized by a cyclic oligopeptide linked with a fatty acid chain, and inside each family that it's possible to observe different isomers that are different between each one by the fatty acid length and the peptides sequence. The antimicrobial peptide produced by Bacillus p34 in previous work carried out by our research group permitted the purification of the antimicrobial peptide and the detection activity, antimicrobial activity against gram-positive bacteria, gram-negative bacteria, fungi, and virus. Phenycin is the main component of the antimicrobial peptide p34, as we're gonna see well explained on my first article. The action of phenycin in bacterial membrane is determined by the antimicrobial and the hydrophobicity represented by the fatty acid chain that is attracted by the hydrophobic core of membrane bilayer. Has been the action against fungi and bacteria has been already demonstrated. So show. And it means to be related with a dose-dependent manner and possibly with a synergic effect with other isomers of the cyclic lipopeptide families. In my work, one of my work I'm going to present you, I considered a biotechnological application, especially a nanoencapsulation of the antimicrobial peptide p34 inside a nanoliposome bilayer prepared with soybean lecithin phosphatidylcholine. And the encapsulation it's consists in the introduction of an active agent inside the whole material. We can talk about nano encapsulation when the particles formed or produced are in a range between 1 and 1000 nanometers. The nanotechnology in food application may help on reduction of the antimicrobial degradation in food preparation, the improving antimicrobial peptide efficacy and stability, and improving of the antimicrobial peptide control release. Here in this slide, I am presenting the workflow I'm going to use on in that article where with the proteomic approach was introduced. A sublethal concentration of antimicrobial peptides were injected in a liquid culture of Listeria monocytogenes during a log phase of growth of bacteria. And then an additional co-culture with the antimicrobial peptide were carried out. This after 1 hour additional hour, protein extraction and trypsin digestion were carried out, followed by mass spectrometry analysis, protein identification, and statistical analysis and discussion of the result. The general objective of my work was study of different antimicrobial peptide responses including biotechnological application on Listeria monocytogenes through a proteomic approach. The specific objects were the characterization and identification of antimicrobial peptide produced by Bacillus p34, study through proteomic analysis of Listeria monocytogenes response to antimicrobial peptide p34, including nanoliposome encapsulation, and the study through proteomic analysis of Listeria monocytogenes response to sublethal concentration of niacin, focusing on the surface protein. The job is going to be divided in 3 different articles, and then partial conclusion and future prospective experiment. The first article was already published by the journal Microbiological Research, and it was titled Diversity of cyclic and microbial lipopeptides from Bacillus p34 related by functional annotation and comparative genomic analysis. And then in the experimental part of this job, I tried to stimulate the secretion of antimicrobial peptide by the Bacillus p34. We have made different co-culture of Bacillus p34 with thermally inactivated cells of Listeria monocytogenes and spore of Aspergillus niger. Aliquots of these different co-culture were used for antimicrobial activity, cell viable con, HPLC quantification of cyclic lipopeptide, and the Malditov analysis and MSMS analysis. In this first job, it was included the genome, the genome annotation and the comparison, and the sequence whole genome permitted to carry out a deeper phylogenetic relation between Bacillus p34 with other group of Bacillus strains. Specifically, Bacillus p34 was included inside the Bacillus velezensis group that is straightly related for similarity to Bacillus amyloliquefacens. And especially here we can see that the much more similar species of Bacillus was the Bacillus amyloliquefacens subspecies Planktorum. This species, since 3 years ago, has been considered the reference species of the Bacillus velezensis group. Actually, also the name is changed nowadays. In this table, I reported the result, annotation result, and of the query sequence and the reference species of Bacillus velezensis group. And as we can see, there is a very similar characteristics between the 2 whole genome. Always using the whole genome, I through the AntSmash software, it was possible to detect different cluster genes that are working for the production of metabolites. And here in this table, I reported also the height similarity with the Bacillus velezensis reference species and the bioactivity detected of each one of these cluster. Here I reported this figure where the different whole genome were clustered together to identify the similarity In the order of the species are from the most similar in the internal part of these rings to the external one that was the less similar. This is the most dissimilar species considering the whole genome. And I reported in the external ring the metabolites identified through anti-SMACE orientation and disposition inside the whole genome. And through this activity, it was possible to identify that two of all these gene cluster were not detected in the Bacillus amyloidosis pattern group, and the Bacillus subtilis used as sister group in the phylogenetic tree, because the species reported here are the same of the phylogenetic tree. In this slide, I reported the cluster genes of fungicine, surfactin, and bacillomycin. That was the main isomers detected in this job, even because they have a higher percentage of similarity inside a different component of the iturin family. The underlying gene represent the core gene for the translation of the enzyme that are co-working for the synthesis of these three main family component. Here I reported a figure where the experimental results were reported. It was possible to identify that there is a synergic effect when there is higher quantity of lipopeptide detected, there is a higher antimicrobial activity. It's related with higher antimicrobial activity. And here I reported the mass analysis result, where it's possible to observe the isomers of fungicin and the iturin ones that were identified such as bacillomycin L and fungicin A and B isomers with different fatty acid length. And here I reported the presence and the absence in the different co-culture created in the experimental part. So in conclusion, we can admit that it was possible with this first job through the genomic analysis, the identification of the strain P34 in the Bacillus thuringiensis group. A considerable amount of fungicide bacillomycin were produced by this strain. The co-culture with fungi spores promoted the bacillomycin production, while the inactivated cells of Listeria inhibited the lipopeptide production. The antimicrobial activity was associated with a fungicin bacillomycin in a dose-dependent manner. The second article I'm going to present to you, it will be submitted. It will be submitted on the journal Molecular Microbiology, and it is titled "The Influence of Free and Non-Encapsulated Fungicin Lipopeptide on Listeria monocytogenes metabolism relevant by proteomic approach." This is the method I'm going to represent all the in both the proteomic study, co-culture, so of Listeria free, Listeria with empty nanoliposome, Listeria with nanoliposome carrying the fengisin, that is the main component of the antimicrobial peptide P34, and the listeria with free fengisin were cultivated in a triplicated, biological triplicate. And after 1 hour of inoculation in this condition, it was possible to carry out the protein extraction and trypsin digestion. Peptide were used for the Orbitrap Veloce mass spectrometer coupled with nanoflow liquid chromatography, and this mass spectrometer analysis permitted to identify 813 protein after elimination of false positive, and parcel D-squared discriminated analysis were carried out to identify the significant protein with different relation, with different regulation, as possible to see in this heat map. All the protein up-regulated were used to create this diagram of Venn, and where it was possible to observe 64 protein up-regulated on the treatment on the treated samples, and the 49 protein down-regulated in this case on the treated samples, because they were detected up-regulated in the control treatment. And to investigate the action of the antimicrobial peptide carried by the nanoliposome, we considered this exclusively regulated protein of protein detected by the treatment with P34 non-encapsulated, and specifically 17 protein were upregulated and 12 protein were downregulated in this case, this group. Two main processes where it was possible to detect considering these two groups of protein. One of the main process was the virulence inhibition process. And here in this table, it's possible to observe all the protein involving in this inhibition process. And first of all, I'm going to present you these two proteins that were downregulated in both the group of protein, so the treatment in the treated samples, coming from the treated samples with encapsulation of antimicrobial peptide P34 and the free action of antimicrobial peptide P34. These three proteins are related with the transported membrane of that is permit to detect the detection of the listeria environment, external environment, and it's possible to this permit the detection. Permit by the other work, it's possible to understand that this protein permit the detection of the host cytosol condition when the PRFA K virulence factor is released and the virulence genes are translated. These two proteins are related with the carbon source uptake and are the main protein upregulated in both the treatment. It means that this protein probably are work and the only way to the carbon source uptake, and they are strongly upregulated. They are part of the PTS system or phosphotransferase system for the fructose uptake. Another important PTS system in this case was downregulated, and this is the PTS system related with the lactose and celluloase uptake. In this case, the downregulation was detected in both the treatment with P34 with treated samples. And just by the protein regulated by the the group coming from the protein exclusively regulated with then antimicrobial peptide non-encapsulated, it was possible to observe the mannose PTS system downregulation. The relation between the PTS system and virulence is related to this in the image that it's showed here. When the PTS system is used as carbon source uptake, the PRFA key virulence factor is phosphorylated to the subunit 8 of this membrane system. When an alternative via of the PTS system is used, such as the glycerol one, as carbon source uptake, the PRFA is released and this is working on the protein virulence translation. An alternative via for the carbon source uptake is the glycerol one, that is the main carbon source system inside the host cells. And here we can see that most of the metabolic enzyme working on the glycerol metabolism were downregulated, especially on the not treated the protein, especially when the antimicrobial peptide is non-encapsulated. Another process that was observed was the metal-ion homeostasis. And here we can see a group of proteins that were related with this process, especially manganese binding, manganese transport lipoprotein that are always present on the membrane of the listeria cells were strongly downregulated in both the treatment with P34 in free antimicrobial peptide. It means this action is coming from the symbols where free antimicrobial peptide were introduced in listeria culture, and the samples were treated with antimicrobial peptide P34 non-encapsulated. To equilibrate this downregulation of all metals inside the cells, listeria cells, a group of downregulated, upregulated protein were observed, especially related with the iron metals uptake. So there is a different way of metal ions uptake by the listeria cells in this condition. So the antimicrobial in conclusion, we can affirm that the antimicrobial peptide P34 permitted to detect two fundamental process targets. The inhibition of the virulent process through the PTS system and the oligopeptide transport that are working negatively on the key virulence factor. Obviously, there are future job in this direction to really understand how this PTS, how the antimicrobial peptide is acting in these two systems. And the downregulation of metal ion manganese for the transport of this metal ion. It's already considered one of the most important antimicrobial targets in gram-positive bacteria for the the specificity for this kind of group of bacteria. And upregulation of the ferric ion metal transport system were observed to replace this deficit of metal ion deficit. And of course, the non-encapsulation exhibited positive support to the process of the metal ion homeostasis and the virulence also. The last article I'm going to present to you, it was published by the Journal of Proteomics, and it was titled Missing Influence on the Expression of Listeria monocytogenes Surface Protein. The method is mostly the same. We just changed the treatment. We used biological triplicate of Listeria monocytogenes and a biological triplicate of Listeria monocytogenes with sublethal concentration of the commercial antimicrobial peptide lysine. And after protein extraction and trypsin digestion, these peptides were submitted to mass spectrometer analysis that permitted to identify 621 protein after a false positive protein elimination. And these proteins were submitted to a partial least square discriminated analysis. It was possible to observe the significant protein with the correspondent regulation on the coming from the symbols of the two different treatments. And here we can observe that it was possible to detect 63 upregulated protein and 96 downregulated protein. Adopting or employing these different bioinformatic tools, it was possible to observe the protein compartment disposition, the protein ontology, and the moonlight protein. From the parting by the 96 protein are regulated, it was possible to observe 18 protein englobed in the group of peripheral cellular component, and parting from 63 protein upregulated, 19 protein were considered part of the peripheral cellular component. We consider for peripheral cellular component all the protein coming from the cell walls and the cytoplasmatic membrane compartment. Also in this case, two main processes were detected. First of all, I'm going to present you the biofilm promotion by the sublethal concentration of lysine. In this process are co-working a moonlight protein and cell mobility protein, and a PTS system for the fructose uptake. Here we can observe there is a downregulation, so there is a different kind of regulation by these two antimicrobial peptide that is working. This downregulation is working positively on the biofilm formation, so promotion. And this biofilm promotion was detected experimentally by a colorimetric spectrophotometric a colorimetric method through a spectrophotometric spectrophotometer detection. And here, before the coloration, it's possible to observe that the lysine-treated symbols presented already, it's possible to observe the biofilm formed. And another important process that was detected in this case was it was possible to observe directly protein involved on the virulence process, downregulated, such as internalin B, listeriosin, or actin A, that are working in this process that permitted the invasion of the host cells. And to co-working with this main virulence protein, it was possible to observe the foldase protein that is working on the folding of the main virulence protein. And then membrane transporter and moonlight protein. In conclusion, we affirmed already that the fundamental action of the nisin on the virulence process and future studies are needed to deep understand how nisin is working on the virulence process. And this maybe can give a light on the listeria resistance to this antimicrobial. And we observed also that the sublethal concentration of nisin improved the biofilm synthesis. And this can be a menace for the real objective of the use of nisin in food industry, because it's important to use that amount of nisin that kill the cells and not working on the biofilm formation, that concentration that is working in listeria on the improving the biofilm formation, that can be a really menace for the food industry. So in the partial conclusion of this framework, it's possible to define that the genome annotation and phylogenetic analysis of Bacillus p34 permitted the identification and inside the Bacillus valens group and the identification of the compound that the microbial produced as phenticin A, B mostly, and bacillomycin L in the reduced quantity. And the proteomic study permitted to define a different response of Listeria monocytogenes to the antimicrobial peptide, specifically the P34 antimicrobial peptide and then the commercial one nisin. And the biotechnological nanoencapsulation of the P34 peptide demonstrated the positive interaction for the antimicrobial activity. Work in progress are the proteomic study of the influence of the enterosin produced by Enterococcus mundi isolated by a buffalo milk on Listeria monocytogenes, and the proteomic study of the antimicrobial peptide P34 free and non-encapsulated on human cells. Here I reported my previous work that I have written during my first year of PhD, and it was published this year on the Critical Review in Biotechnology journal. And then I have co-authored other published and submitted works. I would like to thank this foundation agency, CAPES and CNPq, and the Laboratorio Nazionale di Bioscienza for the proteomic analysis. Obrigado.